Yeah, good morning to all. Now onwards, I will provide the Google form to you. That is a Google quiz. That is a link to you people, so you can put and fill that. Right. So no need to put this continuously this in chat box. So you can have the somewhat relaxed time over there. It. So let me get that link. Yeah, this one is like a one Google link for you people. So within the class time period, if it is maybe remain, you may put in, maybe I will provide a 5 or 10 minutes maximum. After the class, it will be get close. Okay. So what you need to do, you need to click this link and you need to put the required details over there. Let's wait about 1 minute, then we will start our work. Yeah, all right. I think some number of the students are yet maybe remain to join. They will join soon. Okay, even if they have joined late, just I'm putting the links. So now onwards, you just click on this Google form links and put your details, that is a name and your enrollment number over there. Fine. So now without wasting much time, let's continue with our topic. Fine. So yesterday we have started same topic. I'm continuing because it's somewhat larger in a part, but it's very basic things. In the second stage, we'll learn the same points more in detail, and it will be continued in more in detail than after. Okay. So just yesterday we have seen the basic terminology. What is the nutrients and nutrients? Some basic macronutrients and micronutrients, trace elements, and the other sources could be the carbon, nitrogen, then the hydrogen, oxygen, sulfur, and phosphorus are the major event. And the other growth factors are do required for every organism. So it is also included for the bacteria or the microbes itself. Okay. And the basic needs also vitamins is there. So in summary, it was the required the source like energy, electrons, donor, carbon, nitrogen, that is like a nutrients and the metal ions that is important for basic fundamental metabolic procedures. It is like you can say the enzymatic reactions and water. Yes, water is the essential important. You can say the chemical. Without that, the life cannot sustain. Okay. So we have started to talk about the classification based on the source of energy, electron donors, carbon, oxygen requirement, mode of nutrients. So till now we have seen the first two topics, source of energy and the electron donors we have seen. Then we will continue with the source of carbon and the oxygen requirement. Okay, so yesterday we have seen that source of energy. Okay, so basic thing just I'm repeating. Maybe someone is not able to attend yesterday, or it may be required to summarize something. So in that way, the source of energy, that is a phototropes and chemotropes. The photo is a light, chemo is a chemical. that we have seen in their example. Then the second classification was based on the donors, electron donor, that the organotropes and litho, that is the organic and inorganic compound. So based on that, when we are looking for the two terminology to club together, that is electron donors and the source of energy. So we have seen the classification the photoorgano, photolitho, chemoorgano, and chemolithotropes. Till this we have seen. Okay, now we are going somewhat one step ahead. Okay, that is a classification based on source of carbon as an energy source. Okay, so here we are looking at a carbon. Okay, source of what is the source of carbon? So source of carbon, it is like a two possibility. Okay, that is a autotrophs. There we can divide into two autotrophs, and the other are the heterotrophs. They are the organic substrate substances are there. So here it's particular. That is the from the environmental part, they if are able to manage the inorganic carbon, that is a CO2, then they are considered the autotroph, and the heterotroph, that is the organic substrate. That means that autotroph have developed something, it is been either utilized by the heterotrophs. Okay, and there may be certain various mode are there. So here we are going to look at what are they. Okay, so let's dissect our terminology. Yes, we have seen yesterday even. Okay, like a photo, that is that they are using as a uh, energy, that is a light energy. Okay, and the auto, that is we are going to look as a CO2. Okay, CO2 they are going to use for their activities like a cyanobacteria, green sulfur bacteria, and other the list goes on. But the photoheterotrophs are organic substance they are using. Okay, here you can see the green sulfur, here is the green non-sulfur bacteria, and the purple non-sulfur bacteria are there. They are using other than the CO2 as the source of their electron donors. So here the energy is the, we are going to look as a light, but other than the, that is in organic compounds are there. Same it is going with another, that is the chemoautotrophs and the chemoheterotrophs. Okay, so what are the basic difference over there will be here, that a chemical will be the energy. So it may be that it is the some example are being shown here, nitrifying bacteria, right? So they are using the nitrogen. They are using the nitrogen as a source of energy. Okay, and they are autotrophs. Okay, as a chemical they are using. Okay, and they are going to use as a CO2 for it. But chemoheterotrophs, other than the CO2, they can work. So chemoheterotrophs, that means E. coli. They are depend upon the nourishment, the organic compound and their chemical. So sometimes both. Earlier we have also seen this same, right? There is a glucose will be the source of energy and the electron donors. Both could be the possible. The same example we have seen. So here the classification based on the mode will be changing one to another. Sometimes the example is going to remain same. Okay. So one classification is going to describe in that format, another is a different format. So we have to keep in the mind what is being asked at what time, right? Then based on that only we are able to manage and understood the things. Okay, so what was the basic part? The difference, that is a classification that is based on carbon source. So there is a two possibility, autotrophs and heterotrophs. Okay, so autotrophs all are the using the CO2, and other than that it is organic substance, that is a heterotrophs. So what is the basic four classification could be possible? Photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. So photo, that is the energy, is a light. Chemo is a chemical. Okay. So now first step to second step. That is the auto, that they are going to use the energy and the carbon dioxide. Then the photohetero, that is the energy as a light as well as the other than the CO2. Okay, and the chemoautotrophs, that is like some chemicals plus you can say that carbon dioxide, and the chemoheterotrophs is coming that a chemical plus some another compound other than the CO2. Organic compound are there that being utilized into this area. Okay, so to this it is related to CO2 and others. So is the classification we have listed earlier here. So based on the source of energy, we have seen source of electron donors, source of carbon. Now we are going to look the oxygen requirement and mode of nutrition. So this can be a club together. Sometimes you are having the 4 to 6 marks question, right? 4 marks question, 6 mark questions, or the 1 or 2 marks. There could be a possibility. The classification and describe or the differentiate or like that, like source of the energy, source of the electron donor, source of carbon in a 2 marks or 1 marks pattern can be asked. Okay. So many various way to prepare the multiple choice questions could be also possible over here. So you can just keep in that mind the various way to classify the bacteria. Okay, so let's move further for uh, based on carbon source. So before moving, if is it clear that is based on the carbon source classification of any bacteria based on the carbon source, that is the phototrophs and the, you can say, the heterotrophs. Sorry, here we are not using the phototrophs. They are the autotrophs and the heterotrophs. Two classifications are being included. So photo and the chemo, see to two terminology we are clubbing together, photo and the chemotrophs as well as the auto and the heterotrophs. So I hope this point may be clear. Okay, if you have any just questions, you can just put it down in just chat box, or you can unmute yourself. Otherwise, you can write it down the yes, it's clear or the understood, so we can move ahead. Just one based one simple the what we are telling the classification based on carbon source. According to carbon source, can we classify the bacteria? First question. Yes. Okay. So what are the classification? That is the auto and hetero. So we have clubbed together two terminology over here. That is a photo and the chemotrophs. So that will be clubbed together for the answer. Okay, you can unmute also. The rest, that is, these are the some very basic points. Maybe you have learned in your previous somewhere in schooling time or the college time. Yes, I think someone is trying to speak. Okay, if it is there, you can unmute or you can put in a chat box even, so we can have the all together about it. Okay, now let's move for the classification based on the oxygen requirement. It is also an important phenomenon, right? The classification of the bacteria based on the oxygen requirement. Till this, we may have the basic points are the aerobic and the anaerobic. That is the broad classification, right? But in between also there is a some various way to classify based on the oxygen demand. Okay, so let's see about them one by one. So what are they? So aerobic, that is the required oxygen. That is the main important part. Without oxygen, they cannot survive. Another is the anaerobic, that does not require, that do not require the oxygen. If it is like a, you can say that a oxygen lover, oxygen haters. Then some another terminologies are also coming. That is a facultative anaerobic condition that survive with or without oxygen. Okay, that is aerobic and anaerobic. That facultative microbes. Sometimes the basic terminologies are coming as a facultative microbes or the facultative anaerobic microbes are there. So they can survive. If it is the oxygen is there, they will also happy. If it is not there, even they are happy. So what they are looking for the electron transfer in a further different mode. Like we know that the electron transport chain is going on, TCA is going on, but in another directions, if the mode will be there, they go for the ethanol production. So they are the facultative. Then the micro aerophilic, okay, that require the lower level of oxygen. Oxygen is there in their demand, but it is in minute amount. The oxygen, if it is going higher, then it is become a toxic. So here in this classification, if you are moving in the both the direction, okay, So here it is there that oxygen demand is very high, and here the oxygen demand is very low or the zero. So here it is like 100%. Here it is a very scant 0% will be there. 0 to 100 we can classify. So if it is there any amount of the oxygen is available over here, they will happy. And here it is the amount should be as much as lesser. Otherwise, it became a toxic for them to for their growth and development. So here you can see that four that same thing is written. This time I'm explaining that aerobic organism that or the aerobes is an organism that require oxygenated environment. That is a high environment should be rich with oxygen for their survival and growth. While the facultative is that makes ATP by the aerobic respiration, while and but it's capable to switch to fermentation process. Okay, that is like ethanol the productions may happen. So both could be possible. Okay, the basic metabolic process is also can otherwise they can without oxygen they may go for the fermentation process. While the another is like anaerobes or the anaerobic organism that does not require oxygen at all, and it may require may happen that negatively or effect on there. Even the oxygen, if it is present, they will die. Okay, so this is the basic classification, and the yeah, micro aerophile, they require little, very less amount of the oxygen for their survival, but uh, the higher amount of the you can say the other elemental compound could be possible. So based on the here we are looking for the oxygen requirement, how they for survive in that condition. Okay, so classification based on the oxygen requirements. So what is the basic part? That the basic condition is there that some required 100%. Here it is like aerobes. Okay, they required 100% their amount. Here it is a 0%. They does not like. If the oxygen is there, they will say that it is not good for me. And the some are facultative. Whatever it is there, they it is not in demanding. If it is the provided oxygen, they are happy. If the no oxygen is there, they are happy. But some organisms are there, microaerophile, they require scant amount because for the several they may just require the several amount. Afterward, if the oxygen is given more, they will die. And the last is the like anaerobes, they will die directly. Okay, so I hope this point also clear to everyone.
Hello,
Hello,
sir.
sir.
Yes, sir. Same.
Yes,
Yes,
what could be
what could
the
be the
reason for
reason
the
for the
death of
death of
the
the
macro
macro?
microbial like organisms?
Yeah, okay. Generally, what happened? We are going to see in such things more in detail. Anaerobes, there like enzymatic reactions are there. Every metabolic procedure. First, you think about that. We understand maybe a fundamental biochemistry you have learned, or maybe you are going to learn in some other semester. Okay. So every sort of metabolic procedures are happens with the enzymatic reactions. That is a step-by-step catalysis, whether it's anabolism or the catabolism. Okay, that is a breakdown or the joining of the two molecules there. It's conducted by the enzyme. So some enzyme they does not like or the when the sun oxygens are coming in this nearby area, the problem may arise that conformations may change. Either their conformations change or their catalytic activity that is a KM and VMAX, maybe you are knowing or not. I'm not so sure because I haven't seen all your syllabus. So I'm describing in 2 to 3 points together here. That is their basic point is what their enzymatic actions may get disturbed. Once the enzymatic action is disturbed, whole process may be disturbed. One important phenomena. Other phenomena is also there. There is some bacteria having their structural organizations in a way that like a cell wall, cell membrane is there, then they do have certain other covering that is also called the cyst. When the cyst is also disturbed there and the oxygen is reaching to their cell wall to cell membrane, then also their structural organizations may disturb, and that may lead to opening or the closing of the certain channels. So it's depend upon the type of bacteria. Some anaerobes are get sensitivity with their enzymes, some get sensitivity with their structure. So either one it is happening. And yeah, same it is related to microaerophile C1. that if the amount is very high, okay, then it's also that get disturbance. I hope you people get clear about the question.
Yeah. Thank you.
Yeah. Anybody else?
So,
Yeah.
sir, like in micro aerofoil, no, if like the oxygen exceed, and if we are to do the like practical observation, no,
Hmm.
how we can like identify, yes, the like oxygen is getting more, and then the microbes are not able to,
Yeah.
uh,
Okay.
survive.
Understood. Understood. Yes. The third unit, I think I have given some basic explanations related to our whole paper syllabus, right? In the third unit, we are going to learn about that the growth curve. What are the growth parameters are there? And even in our experimentations purpose, I think oxygen related question that is practically there or not. I have to check once again also. So we know that any bacteria, if that is like we can develop such experimentations in a way that you may take the 4 to 5 uh, tubes, glass tubes are there, or sugar tubes you may prepare, and you may start to prepare your solutions, bacterial solutions. Okay, you do have the micro aerophiles in your hand. You have taken about the 10 ml the nutritive materials. Okay, broth, let's take about the nutrient. Broth is there, or the specific organism are there, their chemical demand could be possible. We have taken their broths. Okay, the 5 tubes are there. Let's take the 4 or 5 tubes. Okay, and we have started to put the same amount of the nutritive material, and we have inoculated with the same bacterial suspension, same amount inoculated to every you can say the tube. And now you may start to put these tubes in incubation. One is a control. First you may keep the control, and the 4 tubes are remaining. Now first tube you may keep into a CO2 incubator. Maybe you are knowing about the CO2 incubator and the normal incubator. Let me ask first that question. Do you know about this? Incubators, what is the use of the incubators? Anyone can say what is the usage of the incubators in a microbiology lab?
I think so, like controlling the like even temperature, like contaminations and all.
Okay, contaminations. Okay, we does not want any contaminations in our experimentation. No, okay, even not a problem. Okay, the incubators we are looking. Okay, in incubator there are the two sections are there. So one is the temperature. Another is you can also manage the oxygen demand. That means by the how or the oxygen rather demand oxygen requirements. If you are keeping the plates or the tube on the static condition, okay, that is the platform is given in incubator, you may keep it, so they will stay in that static mode. If you are going to keep flask or your sugar tubes in a rotating mode, so what's going to happen, that oxygen will start to move with the faster rotations via the cotton flux. Okay, via cotton flux oxygens will be reached to the tubes. So one is important is what, their temperature we have control. Another phenomena, that is oxygen requirements, we have solved. So you have kept the first tube into such an environment, that is though they were micro aerosols, okay, that you are knowing, that you have kept into it. Another tubes you have taken, and what you have did in the second tubes, that you have kept in the CO2 incubator. What is the speciality of this such kind of a CO2 incubator is there? Let me say about it. Okay, let me ask, do you know about the CO2 incubator? Have you ever learned?
No much ideas.
Okay, fine. Then let me introduce about it. Like normally, the incubator is like your cupboard, nothing speciality. Or the fridge is there. Fridge is working at a lower temperature, right? And when you are keeping the any particular incubator, so we are keeping the range about like 32, so 40, 45, whatever the range is there. Generally, we are keeping at a 37 degree centigrade. Okay, so it is like a human having that their normal temperature as a physiological temperature is there. In CO2 incubator, the drawers and their glass opening is are specialized in a way that, and what happened, that suction pumps are there in CO2 incubator. So it will suck up all the air outside from the incubator, and it will filled with, and it is fit with the CO2. You can see the gas pipeline is there, like you are knowing the oxygen bottles in corona conditions. We are knowing that oxygen was in high demand, right? So like that, in the we are looking for the CO2 will be get added. Okay. So what going to happen, that one is going to suck, vacuum will be there, the oxygen will be loosened up, and the whole the air will be taken, and it will be fed with the CO2. Now our incubator is a CO2 incubator, and it is required to grow the anaerobic organism. Rather microaerophiles, it is for the anaerobic organism. If you want to grow or the survive for them, then the such incubator is required, which is called the CO2 incubator. So there we have kept. Okay. So aerobic and the anaerobic we can classify based on this. If you are looking for the microaerophiles, so that is a facultative conditions is required in bit. Sorry, not facultative. It is a merging of two. So you can manage the CO2 level. That earlier it is like oxygen maybe 20%, 80% CO2. So such management could be possible to grow such microaerophiles. That is the digitally you can manage. Some are the manual, some are the connected. CO2 incubators are somewhat costlier rather normal incubator. Okay, so it can be digitally can maintain that a percentage amount of the oxygen and the carbon dioxide. If you are looking for to grow in the both these directions, then you need to manage a oxygen bottle also, because you are looking for, otherwise in general condition is what happened in normal CO2 incubator, it's the vacuum up the whole air and the CO2 is fed. But if you are looking to manage this amount of the oxygen and carbon dioxide, so step is what, first is a vacuum is generated, and then the percentage what we have said for the oxygen and carbon dioxide, it will be filled with the bottle. So their requirements of the oxygen will be managed. So I hope it may be clear in a way that okay, now you are looking, okay, sorry, that how we can identify that bacteria that you have isolated, whether it is aerobic, anaerobic, or the micro aerophiles. So what going to happen, that you have grown in the normal incubator. If the bacteria is able to grow, you can certainly say it is an aerobic organism. Where if you have kept in the CO2 incubator, total 100% carbon dioxide, you can say there is a anaerobic organism, and you have set the percentage. Okay, that is why in my this chart, I think, yeah, here I have mentioned or shown in that way only. Okay, that oxygen demand 100%. Okay, here it is oxygen demand is very less, and here it is in ratio. So let's take about the 10 or 15% or the 20%, depend upon the oxygen there, lower levels, but requirements may change. Okay, and here it is, you can look for the facultative, then also CO2 incubator is required, or you can manage with the normal CO2 incubator and the aerobic part. So where our organism is able to grow properly, we can understood the requirement of the oxygen from it. I hope someone has asked, but I don't know the name currently. It is clear to you.
Yes, sir. Clear, sir. Thank you so much.
Welcome, welcome. Anybody else having same questions or related to it? And I hope I'm not going in very much speed. If it is speed is higher, if you find that is higher, you can ask me to go in some lower speed, but it is optimum, because otherwise it seems like that school type. Now you have came in the first to graduation in the second year, right now in third sem. Okay, so basic, you can ask me to increase or decrease the speed. Okay, okay, then let me move further ahead. Okay, even some has where it is the link. Yeah. Those who have maybe joined later, okay, so you can put your attendance in this Google form. Okay, let me move ahead with the next point in the same direction. Yeah, now the classification based on the mode of nutrition. That means what, that like whatever it is, like chemical is available, that is a nitrogen, carbon dioxide, glucose is available with them. Okay, outer side. You have prepared your plate. Nutrient agar plate or nutrient broth is there. You have start to inoculate. So the nutrient is outside. It is like a similar plates, tea, coffee, and the bread jams and everything is lying on a table. Okay, but you need to eat. Then only you are going to get the energy. Or else, if you are sometimes if we are in medicinally or the health is not proper and poor, you are not able to eat. Then what we are doing? Some drinking material is given, or else we are using the some bottles via intravenous. The organic materials or the vitamins and everything is given, or the via injections it is being introduced. So this is the way to introduce the things or the, get the nourishment. So what about the bacteria? How they are getting it? So what is there? The basic bacterial cell is a very unicellular, normal is there. So there is a two phenomena are there. Though that is the osmotrophs and the phagotrophs. That is the nutrient easily enter the cell through the process like a diffusion. Okay, something is like that. Okay, some here the elemental compound are there, so they will start easily diffuse. There is a diffusion procedure is going on. There is no speciality, but the phagotroph is like a phagocytes. So what they are going to do? Sometimes the some material is there over here, so they will start to engulf it. They will enter make a like a mouth like structure. They will develop pseudopodia and the engulfment will be done. The phagocytosis is there. Okay, the osmotrophs and the phagotrophs, the basic diffusion is there, and the phagocytosis. So same part we are going to learn that sometimes even the phagocytosis is not possible. That is like an active and passive transport mechanism there for the material nutrients to be get transferred from one to another organism. How it is there and what is there? Yes, that is in the third point. In same unit, in this our same unit, we are going to learn about the transport mechanism. That is active transport, passive transport, and the diffusion. There we are going to learn more in detail about this point. Okay, so same thing may be written here. Okay, let me put that osmotroph nutrients that easily enter the cell through the process like a diffusion. All the major bacteria are following that category, and even they are looking for the phagocytotic procedures also. They need to put it in efforts like a phagocytosis for getting the nutrition. So majority is like a protozoa. Amoeba, we know is like amoeba, they are developing the pseudopodia and engulfment is done. Okay, so and in osmotroph, the diffusion is there. So it is called the passive transport mechanism. But we are going to also learn about the active transport, maybe not now, but in a third point. So in unit 1, I have distributed or described the whole our your topics into three parts: 1.1, 1.2, and 1.3. So in 1.1, till we have classified the bacteria based on the nutrition's various modes and ways are there, what are there, and accordingly how we can classify them. Okay, so some basic reference books are there: Prescott, Pulsar, or the Steiner. You can refer the some editions, and the some pages and page number also I have given. So same thing you can refer from there, or else you may. I will provide this PowerPoint and other material if required to you people. Okay, maybe that is like maybe they are in 2 to 5 days or within maybe next week, or your attendance and the whole classroom will be set up. So over the classroom, I'm going to upload the PowerPoint and the links related to your YouTube links for this whole entire session that I will upload in your attendance sheet also. Okay, so let me stop here first. What is the time? We do have some time to, we can start further points also. Okay, so anybody having any question related to our topic, just 1.1? Yes, someone is asking for the material first, but I cannot do anything. I'm just following what are the things are here. Okay, there's the basic process and protocols over here that HOD is telling that you have to follow in this format. So they have told me to wait for a while, even told the student about the same. Okay, So as and when the YouTube and the classroom and the everything related to some setup is going on, it will be materialized within day or the by next day. I will provide the material. Otherwise, I think you people asking for the material, okay, but you also able to take the notes. I hope you are writing. Just I'm hopping because I can understand that during the class, if it is in the lecture is going on in the offline mode, people are used to writing down in their notes. I'm not so sure that the same is going on right now. Maybe it is like just you are listening every class, or sometimes you take the screenshot. It is a better way, easy way to get the material or the slides from your professors or teachers. Is it so, or you are making notes even? What is your basic people are following this nowadays? This for in online mode. How you people are just taking screenshot or waiting for the PPT to be get uploaded? Okay, mostly screenshot. Okay, it's become very easy. Okay. Okay, some Yuvraj is there. Have question. I'm taking a screenshot and read from it. Okay, very good. That's good idea. Okay, fine. So seeing what is being talk, the discussion we're going on, you can take it down or not it down. That is good idea. Yeah. Okay. 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 So can you read the last point? Okay. Shall I start the PowerPoint again? But at least you should give us some time to take the screenshot. Okay, maybe a screenshot will require about 1 or 2 seconds, not more than that. And my each slide I'm taking about 4 to 5 minutes. So if you are a slow runner to take the screenshot, I think so. Because the each slide when I'm keeping, it is the whole slide I'm keeping for about, and I'm revising also, same again and again. Okay. Okay, someone is asking then about, okay, let me sort that PowerPoint again. Okay, some questions may be there. So, okay, okay, that is related to your asking. Someone is asking for what? Oxygen demand? What is your question, dear? Yeah, yeah, okay. Rahaf Hashim, so can you return the last point? So what do you want? Shall I discuss again? That means, or what? Okay, that is whatever the thing is there. Just let me introduce. Classification based on O2 requirement is nothing like a aerobes to anaerobes. Some are the facultative anaerobes and the microbes are specifically microaerophilic. That is the oxygen demand. Or sorry, oxygen rather demand. We are going to use the word requirements. Okay, that is sometimes this demand. Why I'm making again and again corrections in this. That is a in environmental science, when you are looking that a BOD and COD will come. I don't know, maybe you have learned about the this terminology or that or not. That is biological oxygen demand and chemical oxygen demand. That will that this terminology will come over there. More or less, it is going to remain same. Okay, that is uh, they are requiring the oxygen does not require, and how much amount. Yes, what we discuss about the fifth. No, no, there till now I have classified till four only, not fifth. Okay. So four major classifications are only there, not much. Yes, Celestina Selema, please, what means what you want to ask? I'm not able to understand. Please be louder. Please. There is your voice is not coming properly. I think. last slide you want to say? So it's going in this way. Yeah, you are looking for this to get the screenshot. Okay. May you share the screenshot of source of energy electrons to make screenshot if you want mind. Okay. Okay, anyway, within 1 or 2 days, maybe by the next week, you will get it. Otherwise, you can just take the screenshot of this reference book. Okay. Or you may just note it down in your diary, some points. You will get the certain information related to it, maybe here and there. Fine. Okay, even if you are looking, then I may just go back. Source of energy. Okay, fine. Let me source of energy. Okay, from here you are looking. Okay, so let me move in that positions. Okay, so you may take the screenshot if you wish. And yeah, those who have remained till to fill this 10:45 somewhere around. So just same slide. Okay, link is there. After 5 or 10 minutes, maybe before about 11 o'clock, I will. Okay, I will close this response. Okay, so you have to respond before it only. No, 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 no. Those who have completed, no need to fill again. Just those who have remained. I'm just telling that those who have remained to fill the their name and details. You can just put it down before the class get over. Afterwards, you are coming that sir, by chance the time was not there and other things, so your attendance will be gone. Okay, let me just put the slide so you can take the screenshot for it. And yeah, if anybody having any questions, also you can ask. So phototrophs, that was a light used as a source of energy. No, no, no. That is, I won't able to do such things for the sharing the material. The thing is, what you can talk to the HOD about the same. What the HOD is telling, I'm doing. Okay. Here the phenomena and the procedures are there that I am following. So I cannot yeah, going to send you an email or an that is in a group or the WhatsApp group, whatever it is there. I'm going to follow the format what is been given to you. I think other faculty members, I haven't seen they're sharing their PowerPoint, right? So what they are doing, they are uploading onto the classroom or the some places are there, their YouTube link. Same I will follow. So again and again, if you are going to some people, students are asking in personally in my WhatsApp also on the same number, that on number. So I'm giving the same answer. If you are going to repeat the same question, it is means you are not listening me carefully what I am telling. Okay, then the classification based on source of electrons, that is organotroph and lithosol. You have got the MB in Google Classroom. Yes, maybe it is there. I will talk with the HOD. What is the Google Classroom and formats? Then I will let you know. Yeah, those people, I am going to note it down even. Those who are looking for the material, I will ask even the same questions. Okay, afterwards. That everyone is looking for the material to be get on their hand. I will even general also quiz. I will quiz for the everything will be there. That I will tell that you got the material 4 days back. That means you are a hurry in a learning, and now you are that is remain idle. Then it will be not good. Okay, if you are asking for the questions, that is get the material on your hand, and that means you have learned it, understood it, and you can ask the questions maybe in 1 or 2 days. But after 1 month you are coming and you are telling that within that if I have the surprise test is there, then you are telling that I have got the material but not got the time. So if you are making the hurry in that way, I will also make some hurry. from my end to get your tested your knowledge. So be prepared for that also. Okay, so I'm giving about yeah, practical could be possible. That I think you were not there in my first lecture when I have introduced. Okay, maybe you are not there. That's why maybe you are asking. Practical session maybe will be if you are coming in the offline mode. If the government is allowing, then we can go. Otherwise, the virtual lab we will go. Okay, so here it is like 10:48, 50 around is there time. So if you are having another questions, you can ask. Otherwise, I'm ending meeting here, and the link is maximum available for maximum 5 minutes. Afterward, that link will be closed for this today's lecture. Okay, welcome to you all people.